Vaccines stand as one of the most significant breakthroughs in medical history. They have dramatically reduced the burden of infectious diseases worldwide. Traditional vaccines have saved countless lives and continue to protect us today, from smallpox to polio, measles to rubella. These vaccines have fundamentally changed human health. They work by introducing weakened or inactive parts of a pathogen, triggering the immune system. This memory allows the immune system to respond rapidly if it encounters the actual pathogen. The impact of these traditional vaccines on global health is undeniable. Smallpox has been eradicated, and polio is on the verge of eradication. Measles, mumps, and rubella are now preventable thanks to widespread vaccination programs. Traditional vaccines remain a cornerstone of public health, protecting millions worldwide. 1. Live attenuated vaccines are a powerful strategy for long-lasting immunity. These vaccines use a weakened form of the pathogen, harmless but able to replicate. This limited replication mimics natural infection, stimulating the immune system. The weakened pathogen trains the body's defenses without causing disease. Examples include vaccines for measles, mumps, rubella, chickenpox, and rotavirus. These vaccines have successfully reduced disease incidence, highlighting their effectiveness. 2. Inactivated vaccines, also known as killed vaccines, utilize a different approach to stimulate immunity. Unlike live attenuated vaccines, inactivated vaccines contain pathogens that have been completely inactivated or killed, typically through heat or chemical treatment. This inactivation renders the pathogen incapable of replication, ensuring safety while preserving its structure. The preserved structure, though inactive, remains recognizable by the immune system, triggering a protective response. When an inactivated vaccine is introduced, the immune system encounters the non-replicating pathogen and recognizes it as foreign. This recognition sets off a cascade of immune reactions, leading to the production of antibodies. These antibodies, tailored to the specific pathogen, act like guided missiles, targeting and neutralizing the threat if encountered in the future. While inactivated vaccines are generally safe and effective, they often require multiple doses to achieve optimal immune protection. This is because the lack of replication limits the extent of initial immune stimulation. Booster doses help to reinforce the immune response, ensuring long-lasting protection. Common examples of inactivated vaccines include those for influenza, hepatitis A, and rabies. These vaccines have been instrumental in preventing these diseases, protecting individuals and communities from their debilitating and potentially fatal consequences. Though unable to replicate, inactivated vaccines effectively present the immune system with a recognizable threat, prompting the production of protective antibodies and providing crucial defense against future infections. Subunit vaccines represent a targeted approach, focusing on specific pathogen components to elicit an immune response. Instead of using the entire pathogen, subunit vaccines utilize only essential parts, known as antigens. This precision targeting reduces side effects and targets specific strains or subtypes. Isolating key antigens minimizes extraneous pathogen material, reducing adverse reactions. This approach suits individuals with weakened immune systems or those susceptible to side effects. Subunit vaccines rely on identifying and isolating specific antigens for a protective immune response. These antigens can be produced in large quantities using recombinant DNA technology. This allows for highly purified vaccines, minimizing allergic reactions or other adverse events. Examples include vaccines for hepatitis B, pertussis, and HPV. These vaccines have been highly effective, demonstrating the power of precision targeting. Subunit vaccines showcase advancements in vaccine technology, offering safe and effective protection. Toxoid vaccines represent another sophisticated approach in the arsenal of vaccine technology. Unlike traditional vaccines that target the whole pathogen, toxoid vaccines focus on neutralizing the toxins produced by certain bacteria. These vaccines use inactivated bacterial toxins that have been chemically treated to eliminate their toxicity while retaining their ability to elicit an immune response. Toxoid vaccines specifically target diseases caused by bacterial toxins rather than the bacteria themselves. By introducing these harmless toxoids into the body, the immune system learns to recognize and combat the actual toxins if ever encountered in the future. The immune response generated by toxoid vaccines involves the stimulation of both B and T cells, similar to other vaccine types. This dual activation ensures a robust and long-lasting defense against toxin-mediated diseases. Examples of effective toxoid vaccines include those for diphtheria and tetanus, which have significantly reduced the incidence of these once common and deadly diseases. One of the primary advantages of toxoid vaccines is their safety. By using inactivated toxins, these vaccines offer effective protection without the risk of inducing the actual disease. However, they are limited to addressing toxin-producing bacteria and often require booster shots to maintain immunity over time. Despite these challenges, toxoid vaccines have proven to be a critical tool in preventing diseases caused by bacterial toxins, exemplifying the continuous evolution and ingenuity in the field of immunization. As we transition from traditional vaccine types, it's crucial to understand the key differences between pre-COVID vaccines and the revolutionary mRNA vaccines that emerged during the pandemic. Traditional vaccines, including those using whole pathogens, pathogen components, or toxoids, require extensive cultivation or purification processes. In contrast, mRNA vaccines deliver genetic instructions directly to our cells, enabling them to produce antigens in situ. The production of traditional vaccines often involves complex and time-consuming procedures, such as growing pathogens in eggs or cell cultures. Meanwhile, mRNA vaccines can be synthesized rapidly using specific nucleotide sequences, allowing for faster development and deployment. Live attenuated vaccines mimic natural infections closely, engaging both humoral, antibody, and cellular T-cell immunity broadly. On the other hand, inactivated, subunit, and toxoid vaccines primarily drive humoral immunity, often requiring adjuvants to enhance their effectiveness. RNA vaccines stand out by stimulating both humoral and cellular immunity through intracellular antigen production, closely mimicking viral infections. While live attenuated vaccines carry rare risks of reverting to a pathogenic form, inactivated, subunit, and toxoid vaccines are generally safer but may be less immunogenic. mRNA vaccines avoid the use of live pathogens altogether, introducing synthetic mRNA instead. This approach brings different safety considerations, such as transient reactogenicity. Understanding these distinctions helps us appreciate the advancements in vaccine technology and the innovative strategies employed to protect public health. The rapid development and deployment of mRNA vaccines during the COVID-19 pandemic exemplify how far vaccine science has come, paving the way for future breakthroughs in immunization. As we delve deeper into vaccine evolution, it's essential to recognize the rich history that paved the way for modern innovations. Pre-mRNA vaccines have a storied past, dating back to the late 18th century with Edward Jenner's groundbreaking smallpox vaccine in 1796. This era marked the beginning of empirical methods in immunization, laying the foundation for future advancements in microbiology. For much of the 20th century, vaccine development was dominated by techniques like attenuation and inactivation. These methods involved weakening or killing pathogens to safely introduce them into the body, prompting an immune response. In the 1980s, recombinant DNA technology heralded a new era with the advent of subunit vaccines, such as the hepatitis B vaccine. By using only specific components of a pathogen, these vaccines improved safety profiles and targeted immune responses more precisely. These traditional vaccines have achieved monumental successes, including the complete eradication of smallpox and the near elimination of polio. Despite their slower development processes compared to mRNA platforms, their efficacy in combating infectious diseases is undeniable. Understanding this historical context allows us to appreciate the continuous journey of vaccine innovation. The lessons learned from traditional vaccine development continue to inform and inspire technologies we see today, ensuring a future where immunization remains a cornerstone of public health. Like, follow, subscribe. We're on YouTube, Instagram, TikTok, Facebook.